Good morning. Can you hear me?
Good morning, sir. We can hear you.
Okay. And can you see the screen?
Yes, sir.
Okay. So, the last topic mm, that I want to cover is another um, modification of the CSTR. These are called recycle reactors, and these are important in two areas. One is wastewater treatment, and the other is recombinant protein production. Um, so um, the reason why they're important in wastewater treatment will become obvious right at the beginning of the lecture. Why they're important in recombinant protein production, where these reactors are actually not known as recycle, they call them perfusion reactors. So most of the proteins made in Europe, these high-end proteins that cost um, I think they call them fancy proteins, right? The ones that cost like hundreds of dollars a gram. These proteins are made using these perfusion reactors, which is just another name for a recycle reactor. But let's just understand the basic idea first. The first question to ask is why do we need them? Well, the reason why we look for something other than a CSTR is that in a CSTR, we must have the restriction D equals mu. That has to hold, right? At steady state, the effluent rate has to be equal to the rate at which you are generating biomass. So, and that mu has to be less than the maximum specific growth rate that you can sustain in the chemostat, given the feed concentration SF. So that means mu has to be less than mu m times SF over Ks plus SF, which we have called the critical dilution rate D sub C. So that means there's a clear restriction on what is the maximum dilution rate you can maintain, and that is determined by this expression on the right hand side. So now the problem is that any system, any set of cells that you're working with, if they have a small maximum specific growth rate, right? So if they grow really slowly, or the strength, the concentration of the feed is very, very small. So if mu m is small or SF is small, then it is obvious that D sub C will be small. And if D sub C is small, that means you have to maintain small mu. And if you have to maintain small mu, then that means you have to maintain small d. So that means you are kind of restricted in the range of d's at which you can operate. Now, this is always the case in wastewater treatment. In wastewater treatment, the cells grow very, very slowly. After all, you're feeding them crap. And compared to the concentrations you use in, for example, when you make ethanol or butanol, you feed them like hundreds of grams. You can feed them at least tens of grams of sugar per liter. But hopefully, you don't have that kind of wastewater, right? The wastewater is as polluted as it is in India. It is nowhere near in the tens of grams per liter, the total organic carbon content. So, in wastewater treatment plants, you would be stuck with a very, very low dilution rate, which means that you would not be able to process large volumes. You would be able to process only small volumes, and that would be a big, big problem. So, um, this is one area where the question then arises: is there any way we can get rid of this constraint, D equals mu? In other words, is there any way we can make D become bigger than mu? Remove this restraint D equals mu by making some design modification. So the idea actually, it turns out, is very, very simple. So what I'll do is actually I will first illustrate the idea, and that is how it was actually shown that it works. but its implementation in the real world requires some modifications because the idea that we're going to study next doesn't work very well in the real world. You have to make slight modifications to make it work. So, so let's start with the mass balance. Actually, before we even do the mass balance, so let's just discuss the principle. This is the principle of recycle reactor. So the principle behind it is really, really simple. If this is your hemostat, with some flow in and some flow out. Earlier, this is some flow out. We're making only one modification. Um, so earlier, so we had, let's say this thing is dipping into the stream. Earlier, basically the effluent was pulling whatever you had inside this well mixed system. This is a well mixed system. You were just pulling everything out into the effluent. So if this flow rate is F, this flow rate is F, this is SF. Here the concentrations are F, S, X, and P, and here the volume is V, S, X, and P. Now the only change you are making is that you imagine that you have put a filter over here. This could be, for instance, right here, you put, for example, a membrane filter. This is the only change you have made to the original chemostat setup. And this membrane filter allows cells, so this allows only cells below a certain size, below a certain size to go through. So, for example, if you take a look at the distribution of cells, if this is cell size, anytime you look at the cells in a reactor, they are distributed. So, this is the frequency meaning the frequency of the cell size distribution. Usually, the cells they're not all of the same size, there is some distribution. And what you have done is you have basically put a filter which makes sure that cells below a certain size, say for instance, only the cells in the shaded red region go through. The other cells which are to the right, which I have labeled in green, these are not allowed to go through, they are retained. And let's assume that because of this, the the so now the picture changes. Um, so now you assume that due to the filter, so keep in mind that this filter is allowing the liquid to pass through the way it was earlier. So now the picture has changed in the sense that instead of this picture over here, what I have is that the flow rate of the cells is, let's say, beta times fx, where this beta is less than 1. This beta reflects the fraction of cells that are allowed to leave. 1 minus beta is retained. They're not allowed to go. There is no change. These are flow rates. And the flow rate of the substrate and product is, of course, unaffected because these are soluble components. They just pass through the filter. So due to the filter, the volumetric flow rate, the volumetric flow rate of cells is reduced, to say beta times F times X. But the volumetric flow rate of substrate and product is unchanged. So basically, this is Fs and Fp, no change at all. So the first thing to see is, so basically, the only change in the mass balances, see that? So the mass balances, for a recycle reactor, barely differ from those are, you have 0 equals, first let's take the case of biomass. This is going to be mu times x. This is the rate at which the cells are generated, which is the same as before. But going out, you have not dx, but beta times dx. The other balances remain the same. This will be ds dt. It was d times sf minus s minus rs times x. Nothing has changed. And likewise for the product, nothing has changed. It is dp dt equals, this is minus dp and plus rp times x. Now, are we clear so far? Are there any questions so far?
It's clear.
Okay, so now you can see right away. So let's call this equation 1, 2, and 3. So the simple modification where you put a filter on the outlet, you can see right away, does away with the restriction d equal to mu, because note that one implies that now you have not mu x equal to dx, but you have essentially beta times d is equal to mu, right? This equation 1, if x is not equal to 0, then clearly you have to have beta d equal to mu, but that implies that d over mu equals 1 over beta, and since beta is less than 1, this is greater than 1. So you basically, by uncoupling the flow of the cells and the liquid, earlier in the chemostat, when you pulled stuff out, the liquid and the cells came together. All you've really done by putting this filter over there is you have uncoupled the two. The liquid is flowing at the same rate, but the cells that are flowing out, you can control them by changing the type of membrane you have. I mean, if the membrane has a high cutoff, you will have more cells. If it has a low cutoff, you'll have fewer cells. So you can control this parameter beta any which way you want. And by choosing this parameter beta, you can make d over mu pretty much as large as you want. So, I mean, there's some restriction as we will see shortly. But the main point is that if you hold back cells, you hold back some of the cells, d can exceed mu,
And it's also clear the more you hold back, the more you hold back the cells, the more you hold back the cells. That is to say, the smaller you make the value of beta, the more D can exceed mu. And in the limiting case where you don't take any cells at all, right? You just don't take them out at all. You can see intuitively, basically, you will never get washout because the cells cannot leave. So that's called like you basically become everything is going back. So it's basically like a batch reactor. You can't get any washout. Everything you make is just recycled right back. Now, the question that we want to ask is, so what do the steady states look like? So what we want to ask the question now is, so we've shown that it's very easy to make D become bigger than mu. But the question is, but what do the steady states do the steady states look like? And again, just like the fed batch reactor, the steady states are intimately connected to the steady states that you can get for a chemostat or a CSTR. I mean, there's a certain relationship. If you know what happens in the CSTR, you can kind of predict what will happen because all you've really done is instead of D, you made it beta times D. So let's start by, but we have to assume. So to answer this question, we have to first, we have to assume some kinetics. I mean, we have to say just what are the values of mu, what are the functional forms of mu, Rs, and Rp. And, we will assume, for the sake of simplicity, so that they follow, we will assume that the cells follow Monod kinetics, which, as you remember, says that Rs is proportional to mu, right? So Rs is essentially, this is the proportionality constant. Rp, the product formation rate, is also proportional to mu. This is why, I guess, Px. And finally, we say that mu itself has the form mu m over s Ks plus s. So this is the Monod kinetics. You know what is happening to mu, and everything else is just proportional to it. Now, if you make this assumption, then returning to this equation here, you can see, let's call this 4. So from this equation, it is clear that so you have mu m, this relationship here basically says that uh, mu m times s over ks plus s is equal to not d, but it is equal to beta times d, right? Then so I should say then the first relationship that is obvious is that mu, which is equal to mu m times s over ks plus s, equals not d as was the case for the CSTR, where it is equal to beta d, but that implies right away that the substrate concentration as a function of d is basically ks times beta d divided by mu m minus beta d. Right? So all we have done is we replaced the formula that had a d by beta d.
So is this smaller or bigger than the concentration in a CSTR? If I take a look at the same dilution rate, if I look at the same dilution rate, will I get a smaller or a higher concentration in a CSTR compared to a recycle reactor? So let's say I have two parallel reactors running. One is CSTR, the other is a recycle reactor. Both are operating at the same D. Which one will give me a lower substrate concentration? The formula is right here in front of you. This is the formula for the recycle reactor. The formula for the chemostat is this one here. Anyone?
So it will be lesser for the recycle.
And why?
So because the factor beta is here and beta is lesser than 1.
Okay. So I think
So in terms of mathematically.
so basically what you have here is let me just show it over here. Mathematically, just take a look at the function. Uh, let's say this is the function Ks times D over mu m minus D, which is actually the substrate concentration in a CSTR. Okay? So let's say at any D, it gives me the value of S. This is S CSTR. Now, uh, according to this, at the same D, um, at the same D, uh, at the same D, at the very same D, the concentration in a recycle reactor will have to be calculated by taking not D, but beta D, which is a lower number. So I have to take, if this is D, so if I take D to calculate the substrate concentration in a CSTR, I have to replace that by beta D. And since this is an increasing function and I'm going downwards, clearly the concentration will be lower. So this has to be less than Ks times D, which is of course equal to the concentration in a CSTR at the very same D. So if I operate them at the same D, I get a lower concentration in the recycle reactor. But we can actually do better. We can actually, so you can get more insight uh, by asking the question. So you can see that from this relationship, which I will call equation 6, okay, so this is or 5. Um, uh, this is, let's say this is 5, and then this is 6. So you can see actually there's a more interesting result um, that if I take equation, in fact, equation 6 implies, so on the left hand side is the concentration of the recycle reactor as a function of D, and on the right hand side I have this expression over here. So you can see that if I try to find out the concentration in a recycle reactor at D over beta, right, that is equal to, I could write it, oh, I see. Mm, okay, so that is going to be equal to Ks times D over mu m minus d. So I'm using this expression over here, simply replacing d by d over beta, and I find that, so and that is equal to S CSTR at d. So in other words, the recycle reactor, so if I have a CSTR which attains some concentration S CSTR D, then that concentration is attained in the recycle reactor at the concentration D over beta, which is much bigger. So you can show this idea graphically, that if this is D, and let's say that my substrate concentration in the CSTR kind of goes like this, okay? And then it becomes, you reach washout. So let's say at any particular value of D, this is the concentration in a C, in, let's say it reaches, this is the concentration in a CSTR. Now this very concentration will be reached in a plug flow reactor at, let's say, basically it will be reached at the concentration in a recycle reactor will always be lower, right? And D can be, um, so basically this is D critical. This is the washout for a CSTR. So I should actually maybe draw it like this. This is the graph for the substrate concentration in a CSTR. And let's say that at a particular value of D, this horizontal line over here represents the horizontal dashed line, which I should draw in red. This one over here represents the concentration of the substrate at the dilution rate D. The relationship above that I have just shown says that in a recycle reactor, the same concentration, the same concentration will be attained at a much higher dilution rate D over beta. Okay? So a given concentration, if it is attained at a dilution rate D in a chemostat, it will be attained at the dilution rate D over beta, which is much bigger because beta is less than 1. So it will be attained at a higher dilution rate D over beta in a plug flow reactor. And in particular, if washout occurs, so what is washout? Washout in a chemostat is when it reaches the concentration equal to the feed concentration. So in particular, if S in a CSTR reaches SF at D sub C, then it will reach S equal to SF at the concentration D sub C over beta. So you have pushed the washout dilution rate also by a factor of 1 over beta. So this graph here represents S in a recycle reactor, which for which I have not used any subscript, but over here I will simply write it down for clarity. So this is the net effect of the uh, uh, so-called recycle reactor. You have pushed the entire substrate concentration curve to the right, but the relationships are really, really straightforward. So if I give you the concentration that is attained in a CSTR as a function of D, I simply have to basically perform this geometric operation to get the substrate concentrations in a recycle reactor, right? I mean, at every point, I just supposing I find out that at D, this is the concentration in a CSTR, then I move that point to the right by a factor of D over beta and exactly put the point there. And that's all there is to it. So this is the simple result for the substrate concentration. Is that part clear?
Yes, sir. It's clear.
Okay, so now the next thing to take a look at. So this is our main result. Now the next thing we want to take a look at is what is the relationship similar that the graph that I have just shown. What can we say about the relationship between the biomass concentrations that you would get in a CSTR vis-a-vis the biomass concentrations in a recycle reactor, right? So this is like uh, performing the same argument now for the biomass concentration. So now, instead of using um, the the balance, here we used to do this analysis, we used this equation, number 4. Now we're going to use, sorry, this is equation number 1. Now we're going to use equation number 2. So looking at equation 2 will tell us about the biomass concentration. So we gain insight into the biomass concentration in a recycle reactor. Reactor by looking at the balance, by looking at the substrate balance, and that balance says that D times SF minus S equals RS times X, but we have assumed RS to be equal to mu over YSX. So it is mu divided by YSX multiplied by X. This is the relationship we have. Now here you have to be a little bit careful. Why? Because this can be easily written as X equals Y. This can easily be written as, you can see, this can be written as D divided by mu, right? Multiplied by YSX multiplied by SF minus S. Now, when we were dealing with a chemostat, we used to immediately put D equal to mu, and we would immediately say, okay, X is equal to YSX multiplied by SF minus S. But now we're dealing with a recycle reactor where D is in fact bigger than mu. And we know how much bigger it is than mu because we said that this quantity is in fact exactly 1 over beta. So that means the final result that you arrive at is this. Okay. Now this is the result that you have obtained for a this is the cell density in a recycle reactor, and we want to ask the question: is this cell density bigger or smaller than the one in a chemostat? So first of all, notice that this we have already found an expression for S, right? Uh, this is actually, maybe I should just write it in the form. Okay. So is this cell density bigger or smaller than the one that is obtained in a chemostat? Keep in mind that the concentration obtained in a chemostat, actually, I should, just to make things clear, I should say that this is X at D and this is S at D. Now I want to compare this to the function ysx sf minus s cstr d. Why? Because this is the biomass level in a CSTR. And what I'm asking is this function bigger or smaller than the function below? Is it bigger or is it smaller?
For
Anybody?
for
So it's
recycle
bigger for
reactor,
the recycle.
It'll be bigger for the recycle.
yes sir.
And why?
Because sir, beta is less than 1 and we are allowing a certain size of uh, cells to pass through the membrane. So there will be more concentration of cells remain inside the reactor.
No, so I mean actually I'm asking for just a mathematical answer. And one thing is that
So the
So
substrate
beta is,
concentration at D is lesser in the case of recycle.
so one reason is yeah, so one reason is that this is obviously bigger than this. One reason because one reason is that S in a recycle reactor at any D is less than S at the same D in a CSTR. That's the first point. So this part alone, the part that I have put a horizontal line on, the horizontal part alone is the one with the overbar alone is bigger than the next term. But furthermore, 1 over, since beta is less than 1, this second part here does a second job on it, right? It makes things even bigger. But by itself, ysx sf minus s of d is bigger than ysx multiplied by sf minus scstrd, simply because scstrd is actually bigger than s of d, right? So uh, it's pretty clear that the biomass concentration at any d in a recycle reactor is bigger than uh, the concentration in a CSTR at the same D. But how much bigger? I think we want a more precise expression. And for that, in fact, um, in fact, oof, so in fact, a more useful way of expressing this is to actually calculate X not at D, but calculate it at D over beta. And if you do that, you end up, you see, with the expression this is equal to, I'm using um, this expression over here. So x at d over beta equals 1 over beta multiplied by ysx multiplied by sf minus s at d over beta. Okay? But we have already shown that s at d of beta, right? This is nothing but s CSTR at D. This is what we showed earlier on, right here. S at D over beta is equal to S CSTR at D. In other words, whatever the CSTR does at D, the recycle reactor does at D over beta, right? That's the expression I'm using over here. But this expression in square brackets is nothing but the value of the biomass concentration in a CSTR. So you get 1 over beta multiplied by X CSTR. Okay? So that means that, and this is at D. Right? This is the result that I have. Now I want to put that in a graphical figure to show you what's going on. So if this is D and this is, I'm using Monod model. So let's say the picture I have is like this in my chemostat CSTR. So this is X in a CSTR using Monod model, which means no maintenance. And let's say this is D sub C, and let's say this is X at any D. This is X CSTR at that D. Then, according to the formula that we have just derived, right, the biomass concentration in the plug flow in the recycle reactor will be something like, we already know that it's the whole thing's jumped. The washout dilution rate goes up. We also know that it is much bigger. We know that it is bigger than the concentration in the CSTR, but we want a more precise result. So this is D sub C divided by beta. Okay. So at D over beta, so let this be D over beta. At D over beta, so this is X recycle. At D over beta, it will be 1 over beta times X CSTR. Okay? So this is the relationship that we got. As you push to the right hand side, so notice the difference. In the case of substrate concentrations, we found that if we move to D over beta, we got the same substrate concentration. But in the case of biomass, so let me repeat that. In the case of substrate concentrations, we found that if we moved from D to D over beta, we got the same concentrations in the CSTR and the recycle, right? That's what this horizontal line in red is showing to you. It is showing to you that the substrate concentration you get at D is also the substrate concentration you get at D over beta in a recycle reactor. Whereas now, if you go from D to D over beta, you don't get the same biomass concentration. You actually get a biomass concentration that is 1 over beta times that in the CSTR. So it's much higher. Higher dilution rates, your biomass concentration keeps on going up. So is that part clear? I mean, and the last thing we want to take a look at. So this is actually finally say the last part is the product concentration, and in this case maybe I'm giving you my notes later on so you can work on it. In this case, the result turns out to be we have time so I can go through it. So now let's consider the product concentration. If we go to the product balance, so from the now considering, and this is where the reason why they use it in the production of proteins will become clear. Now considering the product concentration, so we have considering the product balance, we have dP equals Rp multiplied by X. But we have agreed that Rp, according to our model, is nothing but it's following the Monod model. So it is it is dP equals mu divided by y px multiplied by x, right? But that implies that P is going to be equal to 1 over D, right, multiplied by mu divided by ypx multiplied by x. But x, we have already shown, is d over mu multiplied by ysx multiplied by sf minus s. This I'm using this expression here, this one. We've already shown that x is equal to d over mu multiplied by ysx multiplied by sf minus s. Well, if that is the case, then d and d cancel out, mu and mu cancel out, and you end up with P in a recycle reactor is essentially ysx divided by ypx multiplied by sf minus s. But this ratio is basically equal to ysp multiplied by sf minus s. What I mean by that is, if ysx denotes the amount of biomass formed per unit substrate and ypx denotes the amount of biomass formed per unit product, then the ratio of the two is the amount of product formed per unit substrate. So this actually, YSP is simply the ratio of the two. So that's all we have here. Now, this implies, it follows that P at d over beta is equal to YSP multiplied by SF minus S at d over beta. But this we have also, again, we are trying to find out the relationship between the product concentration in a recycle reactor and that in a CSTR. So that is equal to YSP multiplied by SF minus S. D at d over beta in a recycle reactor is the one that you see in a CSTR at D. And this is exactly the product concentration that you would get in a CSTR at D. If you have any trouble understanding this relationship, this jump here, then keep in mind that just as the amount of biomass formed in a chemostat is YSX multiplied by SF minus S, the amount of product formed in a CSTR is YSP multiplied by SF minus S. I mean, both the product and biomass are products in a single reaction. So if the biomass concentration is given by YSX multiplied by SF minus S, you shouldn't be surprised that the product concentration will have formally the same structure. You can just work it out by going back to the Monod model. I mean, I may not have covered this in the exact formula, but you have the same idea. Do you follow my point? I mean, the model basically says that S dot 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 goes to X plus P. And this is, if you know how to calculate X, you need a corresponding formula for P. It'll be formally identical. P is also like a product, X is also like a product. So this is the formula you get. And in this case, I'll let you work out the thing on your own. Actually, maybe not. The key point here is actually, let me just conclude. So actually, so the picture now is, so this has the same structure. This product has the same structure as that we had for a CSTR. At d over beta in a recycle reactor, you will have the same concentration as the one that you got in a CSTR. And actually, it turns out that the product concentration will look like this in a CSTR, and it'll get stretched out as this in a. So let's say you get this concentration at any D. So this is my product concentration in a CSTR. The same concentration will be reached in a recycle reactor at the concentration D over beta. So let me recount because S and P have very, very similar properties. Whatever happens at a particular D in a CSTR, insofar as the substrate and product concentrations are concerned, whatever happens in a CSTR at D will happen in a recycle reactor at D over beta. But the biomass concentration is a little bit different. Whatever happens in a CSTR at D, right, you will see 1 over beta times that when you move to D over beta in a recycle reactor. I mean, I will show this concisely in a minute, but the main point here is that actually, so this is the concentration. The main point here is this explains why they use the recycle reactor in a to manufacture proteins. So supposing P is your recombinant protein, then you can see that actually you can, by switching from a CSTR to a, so first of all, maybe I should write down the volumetric productivity. The volumetric productivity is actually equal to not P, but it is equal to D times P. Now, if you are able to make protein at the same concentration, but at a much higher dilution rate, D over beta, then obviously your volumetric productivity has gone up, right? So the volumetric productivity in a recycle reactor, right? You can clearly see will be 1 over beta times that you can get in a CSTR, because that is going to be basically equal to D over beta times P at D over beta in a recycle reactor at D over beta. So if I'm operating at D over beta, this is the volumetric productivity I get, and that is equal to D over beta, right, multiplied by P at CSTR at D. So that basically tells you that you get 1 over P multiplied by D times P CSTR. So essentially, the volumetric productivity that you will get by operating over here, the region marked with a red line, yeah, you operate at a higher dilution rate and you get the same product concentration. So obviously, more product is coming. The flow rate at which the product is coming out is 1 over beta times the product, the flow rate at which it was coming out in a chemostat. So you're obviously getting it at a higher flow rate. This is the reason why. This is the reason why. So this is why. So this explains why they call them perfusion reactors. So these are like the perfusion reactors. You know, the $1 billion drugs that they make. You know, these not stuff like Covaxin. I mean, these are like high end, you know, valuable ones. Perfusion reactors are used for production of high value uh, therapeutics. So they just call them perfusion reactors. Now I should mention finally. So let's summarize our key results. We have only 3 key results. The first result is, so key results, these are the ones you need to remember. We have found that S in a recycle reactor at D over beta is equal to S in a CSTR at D. We also found that the product at D over beta in a recycle reactor is equal to the product concentration in a CSTR at D. So, as far as these two things are concerned, whatever a CSTR does at D, the recycle reactor does at D over beta. It's only the biomass concentration that deviates because X at D over beta is not equal to X CSTR times D. It is actually equal to 1 over beta times X CSTR times D. So this should make it easy for you to remember. So you got the idea? Now let me just show you the experimental data. This is the original experiment that was done. This was very long time ago. He actually did it with a membrane. So um, this is from 1970. This is the original paper, and what he did was he used really a membrane filter, and this is the data he got. So he's showing you uh, this is what is obtained. I'm trying to remember now. This is the data with a this is the data obtained uh, with the CSTR. So this is beta equals 1 in our, nothing is removed, everything is allowed to go. And these graphs are for decreasing values of beta. So the smaller he makes beta, that means the more the cells he removes, the further the graph is shifted to the right. You can compare this now with the graph that I drew, and you can see that this is the graph I drew for the substrate concentration. Sorry, that was biomass I was showing you. So this is the biomass concentration. And you can see that the graphs are very similar to the ones we drew. So this is biomass, this is yeast concentration. This is biomass concentration versus D. And here is substrate concentration versus D. This I just showed to you, right? I mean, the idea was whatever the chemostat does at a particular D, the recycle reactor, again, he has done it for more than one recycle reactor. His beta is increasing in this direction. This is the lowest value of beta, then slightly higher value, and this is beta equal to 1. This is for a chemostat. So the theory seems to be in good agreement with the data. So let me finally conclude. I'll ask you to go and take a look at this on your own. The actual implementation of this recycle reactor is not done using membranes. Okay, because these membranes they get plugged all the time, and it's extremely difficult to work with it. So the actual realization, so in reality, so the membrane filter demonstrates the principle, but cannot be used in reality, in real world, because it plugs frequently. It plugs frequently. So the same, I mean, ultimately what we are trying to do is to basically allow the liquid to go through, but the cells are held back. This principle is implemented by essentially two different techniques, in both of which the idea is the same. So I'll explain it with the simple one. What you do is, instead of putting a filter, you actually take out all the cells. You literally take out all the cells. You don't hesitate. You take out all the cells. You carry out the separation over here outside using some technique such as cross flow filtration, which is a very effective technique. There is no plugging. And then what you do is you recycle most of the cells back into the system, right? And only a small part of it. So when you put it through a membrane thing, a thick sludge is formed. The thick sludge is recycled back. The supernatant is released over here, and most of the cells are recycled back except for a small amount. So essentially, this is your membrane, and let's say trying to see how I can. So this is this is the permeate. This is the stuff, that the clear stuff that goes through, um, and this is the sludge you get. I hope that I am doing this right. Yeah. So actually, the picture seems to be bleed, and this is permeate, or this is permeate or cell-free effluent, and this is the recycled stream. So this is the concentrated. By concentrated, I mean biomass concentration is high. Concentrated recycled stream. So this achieves the same goal. I mean, all you've done is basically, instead of uh, holding back the cells, you allow everything to go outside. You do all the stuff outside. Then you allow all of you throw away only a small portion of the cells, which is called the bleed, and all the rest is sent back. So that's just a realization. It's a concrete realization of the membrane filter. Instead of doing it inside the reactor where there are problems, you take it outside, do the stuff, and throw it right back into the reactor. So this, I'll put my notes. You can read it on your own, and I think that we'll take it from there. Are there any questions? I've had to sort of rush through
Just
this,
one
but
question.
uh-huh.
So, in terms of the size of the solute particles that will be present in the cells, could you please tell us the difference between the two, as in the order of diameter of the two?
The solute and the cells. Solute being, for example, the
Yes,
product.
sir. What would be the size of filters that would be required for
Well, I mean,
carrying out the
so
process?
the size of a bacterial cell is on the order of 1 micrometer. So you would, I mean, pretty standard to use, to completely reject the cells. I mean, when you go to the lab, I think you must have used these 0.2 micrometer filters. That's what they use to reject the cells. So if you're working with, for instance, E. coli, the cells are on the order of about somewhere between 0.5 to 1 micrometer. If you use the filter that is, but it's distributed, it is not exactly 0.5, it will be like between 0.2 and 0.75. If you use a 0.5 micrometer filter, you will have rejected half the cells and allowed half the cells to go through. If you did the same experiment with yeast, these cells are bigger. You would have to use a bigger filter. So, for example, this guy has used a filter. He reports it. This is the data that I just showed. Um, he must be using a filter of about 10 micrometers, maybe 3, 4, something like that. So here it is. Uh, oh gosh, he doesn't report the size. He's reporting it in total surface area. So I'm not sure, but it would be on the order, right? I mean, you just the order of the cells. So for bacteria, 0.5 is the order of magnitude of the size of the bacterial cells distributed around that value. For yeast, it's distributed around, 5 micrometers. That would be the size of the filter that you would use. And these filters are so big that they will not restrict the passage of proteins which are on the order of hundreds of nanometers. So they are much, much smaller. Uh, the biggest proteins are on the order of hundreds of nanometers, which would be far too small and would just pass through these filters. Does that answer your question?
Yes, it does.
So if there are any other no further questions, okay. So I will just post the stuff on the notes because actually there's far more to cover, and I guess I couldn't cover everything in the short period of time. It's best to simply put up the notes. You can read it. I just gave you the basic idea, and you can take it from there. I'll be posting some problems on fed batch and recycle reactors later today, and I'll probably also post the problem numbers that you need to solve uh, for the majors. These I will post probably in a couple of days. Uh, okay, so I think I guess I'll see you for the majors then. All right. Good luck and thank you very much.
Thank you,
Thank you,
sir.
sir.